Today we're extremely lucky. We've got Dr. Matthew Caley, who will be presenting his webinar today on models of EB, how treatments are tested before clinical trials can go ahead. Now, Matthew received his PhD in biological sciences from Cardiff University in 2018, studying chronic wounds under the supervision of Professor Phil Stevens. He then moved to Imperial College, joining the lab of Dr. Justin Sturge and Professor Jonathan Waxman to study prostate cancer metastasis and the role of excellular matrix plays in controlling cell behavior. In 2011, he then moved to the Blizzard Institute, returning to skin research in the lab of Professor Idel O'Toole. His research has focused on the skin basement membrane and investigating its role in healthy and damaged skin, as well as in skin cancer. And in 2019, Matthew was appointed lecturer in cell biology. So we're extremely lucky to have Matthew, who's an expert in skin biology and wounds. So before I pass it on to Matthew, just quick bit of housekeeping. All attended cameras and microphones are switched off during the webinar. The webinar will be recorded. Please do not make any private recordings or take any screenshots. Submit your questions through the chat function or the QA function, and please leave any feedback in the chat room to us. And if you'd like to contact us later on, please email us on research@deborah.org.uk. And on that note, I will now pass it on to Matthew.
Thank you, Sagir. So let me just request to share my screen. There we go. So so I've been working on EB for the last 15 years, since I joined Professor Idel O'Toole's lab as a postdoc way back in 2011. I'm going to talk to you about some of the work that we've done and some of the models and some of the ideas we have about preclinical testing. I have conflicts of interest, so I've worked as a paid consultant for Unilever, Partera, and Deborah Research. I'm a board member, a shareholder of Partera, and a founder of the Treat EB CRO. And it's lots of work from that CRO that I'm going to be talking about today. These are my opinions. They do not necessarily reflect that of my employer, which is Queen Mary University. So I don't think I need to dwell on what is EB. It's a very severe and fortunately very rare disease. Its classification as a rare disease is important, and the orphan drug designation is important in terms of clinical trials. There are 4 main types of EB. I'm mostly going to be talking about work on junctional EB and a little bit on dystrophic EB today. We will be starting working on EB simplex later in this year, in the year as some new models become available to us at Queen Mary. One of the important things is that EB, as well as affecting the skin, it's multisystemic. It leads to blistering, it leads to skin itching, it leads to inflammation within the skin. So it's not just as simple as a skin blistering. There's more things that go on, I was going to say, below the skin within EB. So in case you're unaware of how clinical trials work, the clinical trial process starts at the preclinical stage. This is the researcher-led stage, and I'll talk a little bit more about that later. It moves on to phase 1 through 3, where we start with safe dosing, usually in fit and well people, then moving into small-scale testing on patients with disease, and then larger rollout in phase 3, and then phase 4 post-approval is even larger. What you might not know is the amount of time that this can take. So a standard phase 1 trial will take about a year and cost somewhere between £1 and £3 million. Phase 2 is even longer and even more expensive, and phase 3 is long and, because of the large number of patients involved, very expensive. So we're looking at hundreds of millions of pounds to run a clinical trial. Because EB has orphan status, they can do different trial models where you can compress phase 1 and 2 together. So, and phase 2 and 3 together using the same patients, using intelligent study design and using fewer patients than would normally be expected This can both speed up the clinical trial process and reduce the cost, but it's still in the tens of millions of pounds to run a clinical trial. So who pays for these clinical trials? A lot of these are run by industry, by the pharmaceutical and biotech companies that come up with the drugs. They will fund the clinical trials themselves, footing the bill and expecting to be able to recover the cost of that by the sale of their drugs at the end. Within the UK, we're lucky. Our government will fund clinical trials through the NIHR or through the MRC. From 2016 to 2021, when I found numbers, they'd spent over £627 million on rare disease studies within the UK. We're also very lucky within the UK to have charities and philanthropic organisations. I've just pulled out two here. Deborah UK, who have supported 5 clinical trials, with the most recent being ART-EB, the Advanced Repurposing Therapeutics for EB, which is co-funded with LifeArc. LifeArc are, again, a not-for-profit philanthropic charity who support a lot of preclinical and clinical trial testing. The bulk of the rest of the money for clinical trials comes from venture capitalists through what's referred to as venture philanthropy. So this is private individuals staking their own money on rare diseases or on disease clinical trials in general. Again, looking at 2021, the year I got numbers, there was $8 billion invested globally in venture philanthropy. So this is a large amount of money. What all of these funders have in common is that they are very risk averse. Because of the millions of pounds involved in these clinical trials, they want to be certain that they're going to work and they're going to give outputs and give ideas. And a lot of what I'm going to talk to you about today is about how using the right models and asking the right questions is able to potentially de-risk some of the concerns that funders have about investing their money in trials for rare conditions, when there might not be the same ability to recover the investment from sales because of the small population of patients. So I was asked to focus more on the preclinical stage of clinical trials. So this is where we, as researchers in EB especially, come up with an idea, get some funding for the idea, and start doing lab studies. These can be on cells, biopsies, patient samples, and on animal models. Importantly, we don't, at this stage, ever go into patients. If we're going into patients, that's moving into a phase 1 clinical trial, and that is requires an awful lot more paperwork than you need to be able to put drugs onto some cells. So how do we get some of our ideas, and what sort of tools can we use to get those ideas? Well, in my lab, we start looking at patient data sets. We've been incredibly lucky to collaborate with the clinical team at Great Ormond Street and within St John's Institute of Dermatology and the National Diagnostic EB Lab to access 21 years of junctional EB and recessive dystrophic and dominant dystrophic EB patient samples that have been, with patient consent, with family consent, stored for later analysis. So far we've completed RNA-seq. This is a method to look at the expression of different genes within tissue on 45 EB patient samples, and the table expanding that is shown here. So we've looked at 21 junctional EB patients and 22 dystrophic EB patients, with a breakdown of different disease causing genes across the different patients. We've also looked at a couple of EB simplex patients. We've taken those patients. This is from the first batch, but and we've been able to cluster them. And what we've been able to see is that there's actually quite a lot of heterogeneity across the different types of EB. So they have very distinct differences. And when we look at the different clusters, we have the dystrophic EB patients in cluster A, half the junctional EB patients in cluster B, and the other half in cluster C. They cluster separately by laminin, alpha-3 or gamma-2, with laminin beta-3 clustering shared between the two. And when we start looking at the gene expression differences between them, we start seeing differences between the different patient groups. that relate to aspects of the disease, such as wound healing, inflammation, ECM reorganization, that allow us to see differences between them. So what this is telling us is that EB patients aren't just a homogeneous, it's not a homogeneous disease. Each of the different patient groups, and even within junctional EB, we need to be looking at the patients separately because they may respond differently to different drugs. How have we been able to validate these samples? When working with a rare condition, patient tissue is incredibly valuable, and we try and make the best use of any material we're able to get. One of the ways we've been doing that is through multiplexed imaging. We use a microscope called a cell dive, which allows us to stain, image, quench, and then restain the same piece of tissue over and over again. And we've been able to do that to look at markers identified by our RNA-seq. I'll give you a few examples here. So we identified that immune markers were altered, were potentially of interest across all forms of all our EB patients. So we looked at CD45, a pan-immune marker, and here we were able to stain it within our tissue and saw that different EB patient tissue had different levels of inflammatory infiltration. They're all more inflamed than uninvolved skin, than skin from a patient without EB, but they had different levels of inflammation. We're able to drill down into that in more detail, looking at innate versus adaptive immune cells, going back to the RNA-seq data using a method called deconvolution, where we're able to separate out the signals coming from individual cells, identifying that different from the RNA-seq, different patients should have different levels of, for example, T cells, and then going back to the tissue and staining them. So this is for CD4, a T cell marker. Our software predicted that the junctional EB patients with variations in laminin alpha-3 would have an increase in CD4 T cells. And again, looking at the tissue, we were able to see that, so able to validate that. So this gave us some ideas of things that we should be targeting within EB patients. One of them being inflammation. We also identified things like TGF-beta signaling and angiogenesis as being really important and variable between the different EB patient groups. So how do we go about testing that in more detail? So the first step for most preclinical research is in vitro cell models. So we can grow out primary EB keratinocytes. These are the cell of the upper layer of the skin. Here's some little cluster of primary keratinocytes. We can stain the cells using immunofluorescence staining to look at markers. This is a RDB keratinocyte stained with a stain for actin, cytoskeletal actin. And we can do active assays, such as wound healing assays. This is a very simple scratch wound assay, where we can look at the ability of cells to migrate or proliferate. We can also generate more complex models. So I jokingly say that we can grow our own skin in the lab. We do this using a tissue culture insert, a transwell insert. We produce a tendermis. So this is the lower layer of the skin, which is mostly extracellular matrix and fibroblasts. Overlay that with keratinocytes, raise it up so the keratinocytes are in the air, whereas the bottom of the insert is still in the media feeding the cells, and leave it for 2 weeks at the air-liquid interface. And we get these beautiful stratified epidermis, this beautiful stratified epidermis. We can swap in patient cells, or we can make our own patient cells to generate disease models. One of the things that my lab have been working on is how to scale this up. So for preclinical testing, we need to be able to test lots of conditions at the same time. We can now make 80 to 100 of these models in a batch with multiple batches per week. So we can actually do reasonable preclinical testing of drugs or compounds in these skin models. Patient cells are, again, very valuable and can be hard to culture, hard to maintain. So we have taken the approach of using a morpholess cell line. and then using RNA interference, shRNA, to knock down the expression of different genes that are known to be causative in EB in the same cell line, to give us a useful tool for drug screening. Here we've got knockdowns of genes involved in junctional EB, laminin alpha-3, beta-3, and gamma-2. We've knocked them down, tested that by Western blot, and used those to generate 3D skin models. And we've done the same for type 7 collagen, the causative gene lost in recessive dystrophic EB. We've used these in normal keratinocytes and in SCC keratinocytes to give us models to test different aspects of disease. Cell models are great. They are a very useful tool to test new ideas, new compounds, to do that quickly and cheaply and in large scale, but they lack lots of the actual things that we see in the patients. Our cells don't have an immune system. We can add immune cells in, but it's incredibly simple. It'll be an incredibly simplified system. Our cells don't move around. They don't itch. They don't blister in the same way as we see in patients. So we also have access to mouse models of EB. We're very lucky at Queen Mary within our Treat EB CRO to have currently 3 and soon to be 4 models of EB. We have not generated these ourselves. We have rederived them from other labs across the world. So we have an inducible recessive dystrophic EB mouse, originally generated in Freiburg. This mouse, when we treat the mouse with tamoxifen, collagen 7 is deleted, and we can see that by the loss of collagen 7 staining. We have a mouse which has delayed wounding and does generate blisters in the same way as we see within patients with recessive dystrophic EB. We also have dominant dystrophic EB. This is a mouse that was generated in Australia, and we imported into the UK. So this has point mutations in type 7 collagen. It's a dominant dystrophic, so only one of the genes has the point mutation. The other one is unmutated. The mouse is born with mild blistering on its paws and around its mouth, which is exactly the same as is seen in patients with this form of EB. This is a very useful model because it's a point mutation. We can try gene editing tools on this model, as well as looking at treatments that help with removing of misfolded proteins. And finally, we have an inducible junctional EB mouse. This mouse was generated in the lab of Monique Mai in Cologne. We imported it, and we have improved the induction of the model. So we, again, when we induce the knockout with tamoxifen, we get a skin-specific loss of laminin. This is an alpha-3 mouse, so we lose laminin alpha-3. And what we see is blisters within the skin of the mouse, recapitulating one of the key phenotypes seen within the patients. In all 3 of these mice, we see delayed wound healing. So when we do small 2mm punch biopsy wounds in these mice, all of them have a delayed wound healing compared to littermates that have not had a gene deleted or mutated. Again, this is matching with what we see within patients where they have delayed wound healing or impaired wound healing, leading to their the blisters healing poorly. And this allows us to do things like treating the wounds to see if we can accelerate the wound healing. It also gives us a pathway to do gene therapy on the mice, because we can use the wounds as a way of breaking through the skin barrier. We can do other assays on the mice. So an early discovery in our lab was that our mouse with loss of laminin alpha-3 has a barrier defect. So its skin isn't able to be as waterproof as the wild-type littermates. And we can use a tool called to look at transepidermal water loss. This is a measure of skin barrier, and we can see, again, that our junctional EB mouse has an impaired skin barrier, water loss, and we have used treatments to improve the skin barrier on this mouse and been able to record those using the Chul meter. If we look at the patient surveys that Deborah UK have carried out, asking patients across the world what are some of the biggest problems that they face with living with EB, one of the ones that keeps on coming up is itch. And our junctional EB mouse is itchy. So our junctional EB mouse is the one on the right here. Hopefully you can see its hind paw coming up and itching its neck. So we're able to take videos of our mice. They're in their home cages. They're undisturbed. They're just minding their own business while we take videos of them, and we can assess them for levels of itch. And we've been able to look at the amount of sensitive time scratching and how this increases with the severity of their disease. And again, we can use drugs and are using drugs that target some of the causes of itching, which we know is the underlying inflammation within the skin of the mice and of the patients, and we can see how that alters the itchiness of our mice. This is something that's not possible in cell culture or even 3D models because we don't have itching in those models. We can take our mice and we can do techniques such as single-cell RNA-seq, where we take the skin, we break the skin up down to the single-cell level and look at the expression of genes within each of those cells, and we can look across the population. And this is just a sort of snapshot of what we've seen within our junctional EB mouse. One of the most dramatic changes we've seen, again, unsurprisingly, based on our patient data, is changes in inflammatory cells. And again, we see this increase. So in pinky red here are the junctional EB mice, and the green are the littermate controls. And we see this huge increase in inflammatory cells and a lot of B and T cells being increased. We see this in the tissue with the CD45 staining. And again, we see this in the patients compared to some control skin. We see a massive upregulation of immune cells in our models. Okay, so I've spent the last 20 minutes talking about the different models I've got. I'm now going to finish with showing a couple of examples of how we've used our models to look at treatments, preclinical treatments for EB. One of the pathways that unsurprisingly came up in our RNA-seq data was TGF-beta. What was slightly surprising was that it actually came up as being more relevant in junctional EB than in recessive dystrophic EB, which is where it's been more fully characterized. So we decided we would try and target TGF-beta in junctional EB. We first looked at whether TGF-beta signaling was actually upregulated in our cells using phosphorylated SMAD. So this is a canonical downstream protein that's activated by TGF-beta signaling and moves into the nucleus. So nuclear phospho-SMAD is a sign that TGF-beta is active. And we saw increased phospho-SMAD in our junctional EB cells compared to the controls. And these are one of our shRNA model cell lines. We decided we would take a sledgehammer approach to treating TGF-beta, and we've used a TGF-beta-1 inhibitor. It binds to the TGF-beta receptor. It's called REPSOX. It's a very well characterized and very old molecule, and it blocks TGF-beta signaling. And if it's working, we should see a loss of phosphorylated SMAD within the nuclei of our cells. And sure enough, that's what we do. We see, even at low doses of REPSOX, a reduction of phosphorylated SMAD within the nuclei of our cells. And this is significant at all the doses. We then moved into our 3D models of our 3D skin models. So again, in our control and our untreated models, we see this upregulation of nuclear phosphorylated SMAD in our junctional EB in organotypic skin models. and we can inhibit that by adding Repsox into the system. And again, we get nice significances for that, and we've seen this at lower doses. So by blocking by using Repsox, we can block TGF-beta signaling, and that works nicely in 2D and in our 3D models. So we translated this over into our mouse to see if blocking TGF-beta signaling had any effects on the health and well-being of our mice. So we treated our mice topically with Repsox. So Repsox can be delivered by mixing it with acetone and painting it onto the skin. And what we saw was really interesting. So we are not doing anything to correct the underlying disease here. We're not restoring the expression of laminin alpha 3, laminin 332 at all within this model. But treating with Repsox reduced the number and the size of blisters that we see within the skin. So our vehicle-treated mice had lots and large blisters, and the Repsox-treated mice had a reduction in blisters. Even from the H&E here, you can see there are differences within the skin, and these differences related to inflammation. Again, we're not directly targeting inflammation, but when we looked at, again, our inflammatory marker CD45, we see that by reducing TGF-beta signaling, we can reduce CD45 positive cell infiltration into the skin. We also looked at angiogenesis, which is another marker that was identified from our patient data sets as being interesting in junctional EB. And when we looked at angiogenesis, we see increased angiogenesis in our junctional EB mouse compared to the littermate control. And we see that this is reduced by reducing TGF-beta. Repsox is not a suitable treatment for patients. There are other TGF-beta therapies out there. There are also ways of blocking TGF-beta release within the tissue, such as by blocking alpha-V beta-6 integrin, which is one of the main causes of the release of TGF-beta from tissue within the skin. So while this in itself isn't going to lead to a new treatment for patients, because Repsox has many side effects, the targeting of TGF-beta in junctional EB and using alternative methods to target TGF-beta potentially would be useful for patients. And interestingly, we have a platform that we can use to test these in the lab. Finally, I'm going to talk about some work that was supported by Deborah UK and Deborah Ireland. And this is the idea of using protein therapy in junctional EB. So our mouse model lacks laminin 332. What would happen if we just add laminin 332 back into the mouse skin? And again, we can use our wounding model here, where we see delayed wounding in junctional EB as a way of getting the large protein through the skin barrier. So we wound the mice, do a small 2mm wound. In fact, we do two small 2mm wounds. We can treat one with the recombinant protein or the other one with vehicle. And we treat it at day 0, 1, 2, 3, and 4. We can then isolate them and see if we've had any effect on the skin. Looking at the mice straight away, we could see that we had a difference. When we looked at the wound sizes of the wounds treated with the recombinant protein, we could see that these wounds were healing faster. They were smaller, and they just looked healthier. And we could see that when we looked at day 3, we were able to get a nice dose response. So anything over 50 micrograms per mil in terms of concentration of protein delivered to the skin was able to accelerate wound healing. So already macroscopically, we've had an effect on the mice. When we looked at the tissue under the microscope, we were able to see that we have laminin 332 restored to the basement membrane. We can see this in red here in our laminin treated wound compared to the vehicle treated wound. Interestingly, we saw that the laminin actually went beyond the edges of the wound. And when we looked beyond the edge of the wound, we could see that it was actually doing good there. So we looked at blister numbers beyond the edge of the edge of the wound and counted those. And we saw fewer blisters and actually smaller blisters in the treated wound, in the treated protein treated wounds compared to the vehicle controls. Having spoken with companies within this field, one of the sticking points for protein therapy is being able to demonstrate that your protein is actually in the right place within the skin, and it's causing the restoration of specifically hemidesmosomes within the skin. So hemidesmosomes are the structures that hold the epidermis and the dermis together. They're made from transmembrane integrins, binding to laminin, binding to type 7 collagen. On mouse skin, when you do electron microscopy, you can see the hemidesmosomes as these white and dark sort of stripes along the basement membrane. And in our junctional EB mice, these are completely absent. In our PBS treated wound, you've got the basement membrane here, and again, the hemidesmosomes are completely absent. In our recombinant protein treated wounds, we've got the wound basement membrane here, and we're starting to see they don't look as nice as in unwounded skin. We can start to see restoration of the hemidesmosomes. So we've been able to show that using a recombinant protein in our junctional EB mouse model, we could identify a dose of protein that is effective, a minimum dose to deliver to within the skin. We're able to show that we could still see the protein after 7 days. In fact, we can still see the protein present after 14 days. We're able to show improvement within the skin's health by looking at things like blistering, and we see that both at 7 days and 14 days post-treatment. And importantly, we were able to show that we can get restoration of the hemidesmosomes within the skin. At the bottom of lots of these slides, you've seen our logo for Treat EB. So we put all these different models together and made them accessible. So about a year ago, we launched launched this platform. It's accessible to other academics, where we can either share models, share expertise, and allow access to our mouse models, which are particularly expensive to set up and maintain. But it's also accessible to the biotech and pharmaceutical industry. And we are working with multiple partners from within that industry doing preclinical testing of potential therapies for EB at the moment. So that's me at about the half an hour mark. So I've got some quick take-home messages. I've talked about just the models that I have available in my lab. Most labs, most EB research labs have their own models which are very similar. We have this beautiful range of preclinical models to test in EB. What model you need depends on the question you're asking. If you want to look at itch, if you want to look at complex immune responses, if you want to look at wound healing in detail, you really need to be using an in vivo mouse model, and you need to be using the right mouse model, ones that are well characterized. By combining together, as I showed you with the TGF-beta story, the 2D, the 3D, and the in vivo testing, we can get a clear picture of what treatments might work for patients and how they might translate. And all this data together allows us to de-risk potential phase 1 clinical trials and support investment in this clinical translation. Importantly, none of this would be possible without the support of patients and families with EB and within the funders. If you want links to our Treat EB CRO, if you're interested in collaborating with us, please do follow the QR code. I did very little of the work that I've shown you. This was done by members of my group, by my long-term collaborator and co-founder of the Treat EB pipeline, Dr. Emmanuel Rognoni, our long-term clinical collaborator, friend and mentor, Professor Adele O'Toole, and collaborators from across the world who've been willing to share their models, their expertise, and their samples with us. So thank you very much. And these are some of the researchers whose work you've seen presented today. I had the names at the bottom of the slides on a nice sunny away day, getting away from the lab and enjoying some time together. Thank you so much for listening. I'm really happy to answer any questions you might have.
Thank you, Matthew, for an amazing talk. So the forum is now open for questions. So please, if you've got any questions, put them either through the chat function or the Q&A section that we've got on Zoom. So we've actually got a question that's come in. One of the attendees has said they're unfamiliar with the connection between junctional EB and increased angiogenesis. Do we know the mechanism behind that?
No, because I think we're probably the first people to be looking at that. From our TGF-beta inhibition work, it looks to be linked with TGF-beta. This is probably linking back into work I did with Professor Adele O'Toole, looking at TGF-beta, VEGF, and VEGF receptors in recessive dystrophic EB. So probably the same TGF-beta, VEGF pathways that are happening in RDEB are also happening in junctional EB. It was an interesting observation from our patient data set that we obviously didn't expect, and those are the best ones, because then you can start looking at things that aren't obvious and aren't novel. And it was an interesting readout from our mouse. So looking at angiogenesis in vitro is possible, but is much harder than doing it within tissue that naturally produces its own blood supply.
Matthew, I've got a follow-up question on that. It's probably from a position of naivety. I always thought you needed angiogenesis for wound healing, to promote wound healing.
You need the right amount of angiogenesis. So what seems to be happening is there's more an uncontrolled angiogenesis. You're getting more blood vessels forming, more inflammation, more of everything. So if you dampen that down, it seems to be beneficial to the skin. Definitely in RDB patient skin, we see massively increased angiogenesis with very large leaky blood vessels, and that just gets worse if SCCs develop, and then you're feeding too much and allow it to the skin, and you're also potentially allowing transformed cells to escape. So, as with everything, it's finding that Goldilocks zone, not too much, not too little. So yes, you need some, but don't need too much. One of the things that we've observed within the junctional EB mouse skin and within the patient biopsies we've been looking at is this observation of granulation tissue within EB skin. So this is tissue that's highly vascularized, has lots of ECM, not necessarily laid down correctly. It's what you see in the early stages post-wounding that then gets cleaned up, and that seems to be sort of maintained for longer within the skin of our junctional EB mouse, even when there hasn't been wounding.
So just linking on to that, so do you think there's a connection between the amount of angiogenesis and the size of the wounds, i.e. bigger the wound, the more angiogenesis?
Probably, but as I said, interestingly, we see the increased angiogenesis even when we don't have wounds. So yes, I think there's definitely, or when we have just microblisters. I think the interesting question. So if you disrupt the skin, you get an increase in alpha V beta 6, which increases TGF beta release, and we think that the TGF beta is leading to some level to the increased angiogenesis. So yeah, the more disrupted skin you get, the more signal you get, the more blood vessels you're going to get forming around and underneath the skin, I think.
We just had a question that's come through. Is type 7 collagen a good candidate for protein therapy for RDEB?
Yes. But so protein therapy, so collagen 7 protein therapy has been tested. It's gone to clinical trial with Phoenix Therapeutics over in the run over in the US. It's a company owned by Bridge Bio. The original research was done by Professors Mei Chen and David Woodley in California, and it works surprisingly well. They were actually able to show that they could inject the protein into the bloodstream and it would migrate into the skin. That clinical trial, they haven't moved on with the clinical trial because of lack of investment funding. because they weren't able to necessarily de-risk the next stages of the clinical trials. So though it seemed to work really well, it was variable between patients. There is a new company working on type 7 protein therapy called Nova Ancora. That's out of Peter Marinkovic's research group in Stanford. It's a spin out from there. So they are focusing on type 7 collagen protein therapy in wounding, I believe. So yes, it's an excellent candidate. It's a much better candidate than laminin in many respects. Though it's a very large protein, it's a single protein. Laminin needs to be delivered as a heterotrimer of 3 very large proteins. So type 7 collagen is an excellent therapy. What's really interesting is the half-life of type 7 collagen within the skin, so how long it takes to break down. And we've been able to plot this in our inducible mouse. And it takes weeks, probably 20 weeks for it's the half-life of type 7 collagen within skin. So if you get it into the right place, and that's the important part, into the right structure, back into those hemidesmosomes, the protein therapy will last for, or should last for a very long time. So you don't need to constantly redose and retop up.
I have a slightly different question now. And I remember you mentioned earlier that we in WK did the James Linden Lyons research priority setting, understanding what the unmet needs of patients are. And one of the things that came up was itch.
Yeah.
What do you think the mechanism of itch is in the junctional EB?
It's definitely, I believe it's inflammation related.
Mm.
We see this huge level of inflammation, this massive ingress of B and T cells into the skin of our mice, and we see the same thing within the patients. And we know that those cells in other itchy disorders, atopic dermatitis, psoriasis, where you get itch, is linked in with this inflammatory response. So targeting the inflammatory response should reduce itch. And you see that in patients treated with JAK-STAT inhibitors. There's been a small number of inpatient testing of JAK-STAT inhibitors, and they do seem to reduce reduce both the inflammation and the itch. What's important is going to be targeting the right anti-inflammatory to the right types of EB. So what we saw from the patient RNA-seq is that even within junctional EB, not all the patients have the same inflammatory response. So we've been focusing heavily on laminin alpha-3 because that's the knockout mouse that we have. And it's the mouse that works really, really well. And they definitely have a T cell response, but it looks like other forms of EB may have more of a macrophage monocyte response, so wouldn't respond as well to a T cell targeting anti-inflammatory. Even within junctional EB, the patients with gamma-2 mutations or with some with loss of function in laminin beta-3 also didn't have that B cell response. So that T cell, they had a different, the T cell response, they had a different response. So even within junctional EB, 2 patients with the same loss of function, so within laminin beta-3, for example, wouldn't necessarily respond the same way to the same drug because they're having different inflammatory responses based on what we've seen with the RNA-seq, based on what we've seen with our tissue staining of patient samples. So yeah, inflammation is definitely causing the itch, I think, and targeting it, but how to target it is going to be a big question. I know obviously this is something that the clinical trial at King's is hoping to address.
Do you think in the future, I'm jumping ahead of myself here, for junctional EB, you could potentially do co-therapy, so you have some sort of biologic or small molecule that tackles the inflammation, the itch, and then on top of that, you put your protein therapy for the wound closure.
Yeah, I would go as far as also going to gene therapy. So I don't think that, again, looking at the tissue that we've looked at, so all the biopsies, I should have mentioned this, all the biopsies were taken shortly after birth. So these are very young patients, very young skin. They haven't been blistered, they haven't had fibrosis, and yet they're still hugely inflamed. If you do gene therapy on massively inflamed skin, you're not going to get a very good good response, because you're not going to the cells are going are already stressed. They're not going to respond well to your therapy. So doing a co-treatment, treating the inflammation with something, then hitting it with a protein therapy or with a gene therapy or gene correction is probably the way to go. They're much harder clinical trials to run when you're doing multivariants, because it complicates everything. But they're probably the sensible clinical approach, I would say, based on our preclinical data and not having not being a clinician. I'm waiting to get my phone going with some of our clinical colleagues shouting at me about this. But no, for me, it makes sense. You make the environment better, and then you try and treat.
So as a former non-clinical scientist, I'm not going to hold you to anything. So I completely understand that. We've got, it's not really a question, it's more of an ask. Somebody has asked if you can put up your QR code back up for a minute.
Yeah, not a problem. Yeah. I hope it's still working. Let me just re-display and share that. You can also email me. I'm m.caley@qmul.ac.uk. There are two Matthew Caleys out there. I am the one who isn't a poet.
Matthew, while people are taking a picture of the QR code, could you tell us a bit more about Treat EB and potentially how people could get in touch with you to collaborate?
Email us. Myself and Emmanuel Rognoni are basically open to collaborate. So we've been one of the roadblocks to collaborating with universities is always money and contracts. So we have been working very closely with our business development team at Queen Mary, who are superb, and have worked out ways of doing shorter contracts for companies, and ways of to allow us to do cost recovery for using our mouse models for other academic researchers. So we're able to work well with industry through short contracts, and we're able to offer a service to our academic collaborators. So the best thing to do is email us and see what we have available. We are very lucky now to have a dedicated team of researchers who are just working on these models, funded through through Treat EB, with in vivo and in vitro experts in cell culture, tissue analysis. So we can help advise and help design experiments. But with the in vivo one, we offer a sort of a turnkey option. So if you want your therapy tested, send it to us and we'll test it in the mice for you, and then send you skin back. So it means you don't have to try and set up a mouse colony. For comparison, for each of the mouse colonies we have, it probably took us over a year to establish them properly for each of them within the university here. It's extremely time consuming, extremely expensive, and doesn't need to be replicated when you can use ours. So that was sort of the plan, the goal for this this model, to make it easier for other people to do research.
Yeah, we've just had a question come through. Is dominant dystrophic EB a possible candidate for genetic/protein therapy?
Yes, very much so. So one of the main reasons we have the dominant dystrophic EB model is to practice doing gene therapy on it. The problem with skin in gene therapy is that skin is a really good barrier. It stops things going into it really, really well, which is why we are all comfortably, we're able to go swimming, we're able to wander around the streets without without issues because our skin barrier works. And even impaired skin with blisters, with holes, the skin barrier still holds and prevents the delivery of most gene therapy, most methods for delivering gene therapy going through it. So one of the reasons we wanted to have this mouse and we're using it is to practice how to do gene therapy in EB skin. How can we get things through the skin barrier to the keratinocytes, the basal keratinocytes, which are the ones that we need to correct the stem cells or the progenitor cell populations there that will allow for permanent correction. So yeah, very much a good candidate for gene therapy. In terms of protein therapy, we are testing that at the moment. Some of the problems depends on the mutation of the variant. So some of the variants cause the protein not to fold properly, and it sits around within the cell and causes issues there. This is particularly a problem within EB simplex as well. Misfolded keratins sort of clogging up the cell. So protein therapy by itself wouldn't necessarily be as beneficial. However, other therapies that are able to clear misfolded proteins, target those, things like mRNA antisense therapies are actually perfect for dominant dystrophic EB or for EB simplex, depending on the variant. This is why having the right model and asking the right questions is so important, because it changes the treatments, it changes the method of delivery.
Thank you very much, Matthew. There aren't any more questions coming up. So I think on that note, I will wrap up the webinar. So again, Matthew, thank you very much for an engaging talk today. I've certainly learned a lot today about your mouse models, and especially EB-Treat, which seems to be an amazing platform to test lots of potential biologics or small molecules or therapies. So if anybody out there's listening, please get in touch with Matthew. Or
Emmanuel Rognoni.
Or Emmanuel Rognoni. Yeah.
Flood our email inboxes. This is all fine.
Please do that. And on the note, I will come close this webinar, but just a highlight, please, if you haven't already done, register for our June webinar, which will be at a later time of 8 p.m. because we will have Dr. Emily Gorrell, Assistant Professor of Dermatology from the University of Colorado, hence the time difference. And she will be giving an overview of gene therapy in recessive dystrophic EB. So please, if you haven't joined, do register for it. And on that note, once again, Matthew, thank you very much for today's webinar.
Absolute pleasure. Thank you so much.
Okay.